Welcome Genetic Teacher channel. Today, we are talking about viable bacterial cells counting, which used to measure effects of bacterial growth, using spread and pour plate methods, filter membranes method, and serial dilutions methods, to distinguish between living and dead bacterial cells. As we showed, spread method, initiated by inoculated solid media with 0.1 milliliters, bacterial dilution, then spread inoculum over solid media evenly, to allow bacterial colonies to grow only on surface media, while pour plate method, initiated by inoculating 1 milliliters, or 0.1 milliliters, of bacterial dilutions in empty plate, then add melted nutrient agar above inoculum, and swirl, to mix, to allow bacterial colonies to grow in and on solidified media. Meanwhile, in filter membranes methods, bacterial cells retain in filter membranes, then transfer filter membrane to culture media for inoculations, to allow bacterial cells to grow in form of colonies. Whereas, in serial dilution method, bacterial samples diluted 10 times, by adding 1 milliliters, or 1 gram, of bacterial sample, to 99 milliliters distilled water, as mentioned inside slide. Thanks for attention. Please like and share and subscribe Genetic Teacher videos and channel.